Good morning to all. So yesterday we are discussed it is about the transcription and the translation process. So at the end of our translation process, what happens is it is mainly the structure of that was a mRNA strand here. So in these 5' into 3' and in these reaction. So it is a P site, A site and E site here. So here are the codons and anticodons of mRNA are going to bond with the tRNA. So it is a peptidal tRNA place. Peptidal tRNA. So P means peptidal tRNA, A is the it was the accepting stage, and the first stage that was a fresh amino acid is first going to be occupied the place of A site. So that was mainly after reach the amino acid and the stage of these A site, then immediately that was going to be transmitted to the peptidal tRNA. So in these peptidal tRNA, the amino acids are going to bonded with the anticodons. So tRNA may contain the anticodons. So which mRNA coded the information from the DNA. So those coded information was going to be bonded with the tRNA. So anticodons, the codons which are presented on the surface of mRNA. So those are bonded with the anticodons of tRNA, and both are tightly bonded with each other and form the amino acids here, the methionine. So in this way the amino acid chain was continued, and a fresh amino acid was reached here, that was transmitted to this region, and immediately that was bonded with the peptide bond, converted to amino acid, and through the exit site that was released out. See this. So in this manner, that was all the amino acids are entered and transmitted to middle part, and so through the exit region, so all are got exist. So that was the from that region, so those are exited and released out. So with the help of releasing factors are also there. So releasing factors. So with the help of these releasing factors, so amino acid chain was prolonged like this. So in this manner, in this direction, the amino acid chain was prolonged. So at the end of any stop codons, UAA, UG, UAG and UGA. So these three codons may not be going to be code any amino acid, and these are considered as a nonsense codons and also considered as a stop codons. So the amino acid receiving so that was occupied the A site. So these three amino acids, any of these three amino acids are got reached to this region, then immediately what happens is the translation process was going to be stopped completely. The translation process was going to be stopped. So that was a termination process, and these are also considered as a terminator codons. These are also considered as a terminator codons, means the process of translation was going to be terminated with the help of these three amino acids. So those are nonsense codons and also considered as the stop codons. So these may not be going to code any of the amino acid, that's why these are considered as the stop codons, terminated codons, and also called as nonsense codons. So at the end of translation process, it was takes place here. So when these three of any one of amino acid is reached to these A side, then immediately the chain, amino acid chain, that was a polypeptide chain, means many amino acids are fused to form, that was considered now as polypeptides. So many numerous amino acids, so those chain was deviated from the tRNA. See this, that was completely separated and free like this, and these amino acid chain, polypeptide chain was reached to the destination part. So and again the transcription again started, the process was repeated for many number of times. So the process was repeated and the amino acid chain was separated from that peptidal tRNA. So this process was a transcription and the translation. And now today we are going to discuss about the genetic code, that is a gene expressions, that is the genetic code. Today we are going to discuss about the new topic, that is a gene expressions genetic code. Gene expressions, those are going to be so converted and coded the information, so that was going to be expressed. So in the way of so those are coded the information which was got from the DNA. So that was the information inside the DNA was got so received by the mRNA in the form of a coding mechanism. So those coding mechanism was mainly received from the DNA to mRNA. So those coding information was presented on the surface of the mRNA. So that was again finally reached that information into the tRNA. So the tRNA may convert so those coded information into the so that was amino acid chain with the help of bonding with the amino, that is anticodons. The codons which are presented inside the mRNA, those information came from the DNA. So that was the coded information was going to bonded with the tRNA. So these tRNA was going to convert those information into the amino acid chain. Finally polypeptide chain was formed. So these this type of mechanism was considered also as gene expressions. These are also considered as the gene expressions and the genetic code. So genetic code, so that is a relationship between the codons and amino acids. Codons and amino acids. A relationship between the codons and the amino acids are so that was considered as a genetic code. That was used to considered and so the called as a genetic code. The definition of genetic code was a relationship between the codons and the amino acid. A simple form, codons, those are came from the DNA. So the information, the hereditary information which is presented inside the DNA was received mainly by the that was the mRNA. So that was stored that information in the form of codons. So those codons are going to bonded with the so anticodons which are on the surface of the tRNA. Both are bonded with each other and finally convert an amino acid. So that was the conversion between the codons, the coded information which is presented on the surface of the mRNA with the tRNA anticodons. The codons are going to bond with the anticodons of tRNA. Then this type of relationship so was considered as that was called it as a genetic code or gene code. So these are the genetic code. So the mainly it was a triplet codon. Triplet codon means so that should be contained the UAG. So that was a so mainly the methionine is the initiator amino acid. So UCG. So UGG. So in this manner, so any one of the amino acid, totally 20 amino acids are in there in the environment. So these amino acids are going to bonded with the 64 codons, means each one of the amino acid is going to bonded with coded with the 3 genetic codons, 3 codons. So those are codons. So the 4 some of the so 3 or more 3 or some 2 in the middle of 3 to 4 amino acids are also going to coded with the more than 3 amino acid, that was 4 amino acids also. So the 4 4 codons, so 4 genetic 4 codons. So actually each and every amino acid is coded with the 3 of the genetic codons only, but so some of the exceptional cases are also going to, so UCGU. So here are the 4 codons are there, but it was going to consider only 3 codons are considered here. So these extra one may not be going to be considered. So in this manner, some of the 3 or 4 amino acids are also going to so coded with the more than 3 codons means up to 4 codons. So that's why totally 6 4 codons are going to code the 20 amino acids actually. So 20 amino acids are going to code with the 64 codons. Some table is there, the genetic code table here is. So it was started from here. see this. So it is a genetic codon, the genetic codons table. So actually, so amino acid, 20 amino acids are there. So on those 20 amino acids, and some of the amino acids are also repeatedly coded with different types of the codons. That should be also occurred here. So you can able to observe here. So that was mainly the UUC, UU again. So it was a UAC and UUA. So these three are coded for phenylalanine, and UUA and UUG are leucine. It is also one of the amino acid, and the serine was coded with these three different types of the codons: UCU, UCC, UCA and UCG. And so tyrosine, so that was UAC, U, and so that was the again UCA. So these two are for tyrosine, and UAA, UAG are so not coded for any amino acid, and as these are considered as a stop codons here. So UGA is also one of the stop codon, and remaining UGU, UGC, UGG. So these two are for cysteine, and UGG for the tryptophan is one of the amino acid, and leucine. Here the leucine is coded for these UAA, UUG, but so these four are also coded for the leucine. So those are the CUU, CUC, CUA, CUG, and proline. The proline was a CCU, and triple C, CCA, CCG are proline, and CAU, CAC, CAA, CAG for histidine, and so that was another one is a guanine, and so CGU, CGC, CGA, CGG for arginine is one of the amino acid, and isoleucine, that was AUU, AUC for isoleucine, and the AUA and AUG. So it is a methionine is the initiator codon, initiator amino acid always, and a threonine. So it was the threonine. ACU, ACC, ACA, ACG 4 are coded for only threonine, and aspartate AAU, AAC, and AAA and AAG for lysine, and so AGU, ACG, AGA, AGG for serine and arginine, and valine. So that was GUU, GUC, GUA, GUG for valine, and GCU, GCC, so GCA, GCG for alanine, and GAU, GAC, GAAA, GAG for glutamine, and the aspartate is GAU and GAC aspartate. So another amino acid, and the GGU, GGC, GGA and triple G for glycine. So here as the guanine, and so different amino acids are coded here. So those are lysine, leucine, isoleucine. So these are the different amino acids, and repeated amino acids are also coded with different types of the codons are also available here. You can able to watch here. So those are so sometimes they coded for one of the amino acid, and some another times those codons are coded for the so same amino acid with was coded with different types of the codons also occurred. So see here leucine here is presented, and the leucine in somewhere is also presented, but these are so for any so these two and these two, and these remaining four completely are coded for a leucine amino acid, means so those are complete, the combination is different, but still so those are coded with the only one unique amino acid, means so their codons are vary, these codons are vary, but they used to code a single amino acid. So those also happened. So phenylalanine, leucine, isoleucine, methionine, valine, alanine, threonine, proline, serine, tyrosine and histidine. So so glycine, glycine and lysine, so aspartate, glutamate, cysteine, tryptophan, arginine. So these are the different amino acids are going to coded, and so any, so see here UGA, UAG, UAG. So UGA, UAA, UAG. So these three are completely stop codons and nonsense codons and terminator codons of translation process. Why? Because these three are not going to coded with any one of the amino acid. So that's why these are considered as a stop codons, end codons. So this is a table for genetic code. and we are going to discuss now the properties of genetic code. Now the properties of genetic code. So those properties are, first one is comma-less, means so in the mRNA 5' to 3' end, so AUG, so that was AGG, ACG. So in this manner, so that was continued, and on the surface of the tRNA also the process was continued with a one by one amino acid, but there is no comma, there is no full stop up to they reach to the stop codon. Up to this reach to the stop codon, there is no comma, there is no full stop. That's why the property is comma-less. And so the genetic code is a universal, means so each and every organism, so completely in the universe we can use a unique genetic code for each and every organism. So that's why that is a universal. So each and every organism, these amino acids are completely, the codons are may not be going to be changed. So that's why, so that is there is no comma, full stop, that is a comma-less, and it was completely used by the overall the universe. That's why it is a universal. And degenerative. So means so it was completely at the end of so each and every so amino acid was coded with the 5' into 3' direction. So in these direction it was continued till to, so that was reached the tRNA on the surface translation process. They reached up to the end of stop codons, up to till that region that was not going to be so degenerated up that so it was a degenerative process. So then they reached the stop codons, then only that was going to be stopped, otherwise that may not be going to be stopped. So that status is called as a degenerative status. That is also one of the property. And polarity is one of the process. So polarity is one of the process, that is 5' into 3' direction only. So always the chain, the strand of mRNA, and so that the synthesis of mRNA was also continued in the 5' into 3' direction only, but not in 3' to 5' end direction. So but that reached the 3' end direction, what happens is the amino acids, the codons are presented on the surface of the mRNA. So those are bonded with the anticodons of tRNA. So at that time, so that what happens is on the surface of the ribosome, the tRNA is a middle part, that is peptidal tRNA is a middle part. So the occupied place, that is A site, and middle the P site is there, and the last site is the exit site. So on this way that was moved in somewhat 3' into 5' direction, but at the time of the formation of bounded with the codons with the anticodons, so at that time only that should be happen. So otherwise that may not be going to be happened. So that situation. So that's why the properties of genetic code, that is a comma-less, there is no comma, there is no full stop up to till the end of that and it was a universal and polarity. So and so these are the some of the properties which are going to be performed by the genetic code. So it is the, actually see this, it is the DNA, and from these see here, so it is the DNA. From these the information was to the mRNA, that is the transcription, and from that to, so mRNA, DNA to mRNA is the transcription process, and so from that to the information was reached to the single stranded mRNA, that is 5' to 3', and it is the 5' to 3' and a 3' to 5', double stranded, it is only single stranded, and here mRNA to tRNA, so that is a tRNA to the protein, amino acid chain is formed. So this process is considered here as the translation. So the tRNA to protein, that was mainly occurred in the middle of, that was the codons AUG and UCG and UGG. So these are the mRNA strand. So these was continued in the 5' into 3' direction. So these are the codons which was received the information from the DNA. So these process was also considered as central dogma and the main phenomena with the genetic code. So those are the some properties of our genetic code. So today's our topic, that is genetic code, is completed. So we'll meet tomorrow. Thank you.